Welcome to Channel Today in History. In today's video we will talk about the day, October 25th, first cloning of human genes, 1993. The word clone has acquired new uses as science advances and is applied in the form of technology. Initially, it was used to designate a population of cells or organisms obtained by vegetative, asexual, reproduction from a single cell, so that all members of a clone have the same genetic constitution. Later, when genetic engineering made it possible to multiply a gene or a piece of DNA from bacteria, the term was extended to gene cloning. In higher animals, it was impossible to apply this concept, as they could not reproduce asexually, so to clone them it is necessary to surgically remove the nucleus of a fertilized cell, zygote, and replace it with the entire nucleus of another one, animal. The first experiments of this kind were done with amphibians. Toad eggs were chosen, as they are a large cell, easy to obtain and manipulate. Their nucleus was removed and, on the other hand, the nucleus of embryonic cells that were still totipotent cells in an early stage of development that can derive into any cell type, which were introduced into enucleated frog eggs. Finally, these studies were relatively successful, and it was possible to create cloned frogs, identical to each other, with identical genetic constitution. When an attempt was made to introduce already differentiated cells from adult tadpoles or frogs, the experiment failed, and the resulting embryos did not live long. This study led to the discovery that cells already differentiated were incompatible with the cytoplasm in which they were implanted, and that the resulting nucleus was unable to replace that of the embryonic cell. In 1952, the cloning of frogs was successfully achieved, which left latent the possibility of taking the same step with mammals, starting from an adult animal, a challenge that led to the creation of several research groups that tried to clone rats over the years 1980, but the failure was complete. University of Edinburgh researchers led by Ian Wilmot managed to overcome this hurdle after 40 years of exhaustive research in genetics and biology of reproduction, reinforcement in embryo manipulation techniques and assisted reproduction, and multiple experimental trials, until finally obtaining the sheep dolly, the first mammal cloned from an already differentiated adult cell. The cloning of higher animals and human embryos had previously been possible, through a different technique, where blastomeres were separated from embryos in a state with about 8 cells before they differentiate, obtaining identical embryos with the same constitution genetics. In 1993, George Washington University researchers Jerry Hall and Robert Stillman succeeded in cloning human genes and later released data from twin incision experiments on human embryos of 2, 4, and 8 embryoblasts they performed themselves, but these embryos never were transferred to the mother's womb, due to the ethical connotations that this implied. This discovery was within a series of experiments that did not have the prior consent of the competent ethical committee, causing agitation and alarm. Recently, other researchers have expressed their opinion on the need to guarantee research freedom. Did you like the video? In the description you will find the blog link with the full article. Give an incentive by subscribing to the channel, liking, commenting, and sharing with your friends. Hugs. Until the next.